Okay. Well, it is just after 4 o'clock. Thanks, everybody, for coming. It's a pleasure to be able to be here and talk with you about some of my ongoing research here at AOML. It claims to be. Can you folks online hear me? Ryan Smith, you want to type a yes? Anyway, well, we'll pretend that, yes, thank you, Lisa.
Yes, we can hear you.
Oh, hey, thanks. All right. So today I'll be talking about environmental variability and the movement ecology of marine organisms. Or so it seems. So to get things started, I'll explain what it is that I mean by movement ecology. And I'll talk about the importance of putting organismal movements in an environmental context, thinking also about the importance of the behavior on the movement of animals and other organisms, and then get into some of the applications and emerging research areas related to organismal movements. So first, movement ecology is a relatively new field. It was pretty well described in the mid-2000s, sort of putting together a conglomeration of sort of these disparate research approaches under one roof. And so what its aim is to be a mechanistic approach for studying the causes and patterns of organism movement. And this has been really driven home by a group in Israel, Ran Nathan, who is leading this in some ways, at least initiating it. And in any case, what the aim is to understand sort of the movement path of organisms. So a step-by-step process which is influenced by these factors, which are also interact with one another. But the idea is that if you understand the external factors that are influencing, say, the internal state of an organism, which gets at the question of why move. So the internal state might be, is the animal hungry? Is it scared? Is it big? Is it sexually mature? Those sorts of things. So you have these external factors that might influence the internal state, which also influence the navigation capacity, so where to move. Is it, and that might be both sort of an orientation question or a timing question in terms of should I go left, should I go right, is it, should I go up, should I go down, and how it is that they know sort of which way is right or left or up or down, those sorts of questions. And then these things feed into the motion capacity, how to move. So this might be sort of on the biomechanics side of things. You could think about different gaits of organisms moving. You could think about the sort of the physiology of the animal, how fast it can go. Is it is it passive, perhaps? Is it, for instance, maybe like a seed that is dispersing, or is it capable of sustained motion? So each of these four things sort of converge on one another and lead to a single step of an organism. And then they get reorganized and you have another step, all of which keeps feeding back into this loop. And so what you have is something like this, where you have movement steps of an organism, these different phases of movement, and a lifetime track of an animal. So all of these pieces can be studied from that sort of mechanistic perspective. And by it being so focused on sort of the individual steps, or the steps of individual organisms, it can be particularly useful in a modeling perspective, and can be sort of extrapolated and applied out by, you have this individual moving. If you sum a lot of individuals and their movements. What that gives you is sort of this time-varying population distribution, where if you sum these across an entire population, then what you can get is sort of you could take slices and say at time 0, what is the population distribution? At time t plus some number, what is the population distribution? And you know what caused it. If, we'll go back a step, if you have gotten these things, you can then make predictions about how, as external factors change, what will be this result for the individuals, and then how that will sort of scale up to have these population level implications. So, what my aim is to sort of blend a combination of theory and empiricism, so getting data, merging that with a modeling framework to fill in some of the knowledge gaps in movement ecology, which I'll discuss. Maybe I'll hit on that sort of at the end of this talk, actually. But in any case, what my aim is to take these disparate pieces of data, say lab-based behavioral experiments, environmental models, tracking data of organisms, feed the information that we get from each of those things into movement models, which can then give us insight into these population level sorts of questions, sort of like I discussed already, the distribution of populations, and then also multiple populations. You can start thinking about connectivity as you overlay these movements on top of, say, our own, let's say fisheries locations or energy extraction locations. We can start thinking about how anthropogenic interactions might occur, and then do things like set conservation priorities. And if we're able to get good links between sort of environmental conditions and behavior and how that relates to movement, we can start to get inferences about things like how populations and maybe even ecosystems will be responding to global climate change. So in all of this, what we see is that the biology and the physics should be of equal consideration. It's not to say that they are always of equal importance for a given movement step, but it certainly goes hand in hand, which might make sense. You have an organism, it's biology, it's moving through a fluid, for instance, say the ocean surface, and those movement steps that it takes is going to be a combination of its swimming velocity and the velocity of the surrounding fluid. And then, of course, with this simple schematic, if the swimming velocity, if this arrow is relatively small compared to this arrow, then perhaps the current will dominate its movement. By contrast, if the current arrow is relatively small compared to, say, the swimming, then the behavior of the organism will dominate its movement. And this will probably vary at different times of an animal's life. As its swimming capacity grows, it will vary depending on the species that we're talking about. That current arrow would be much more important for perhaps a small fish larvae than for a whale. But in any case, looking into it from this framework, we should be able to have sort of a unified theory for addressing organismal movements in the marine environment, sort of irrespective of taxa. So that's one of the things that in particular I'm aiming for. And in doing this, one of the things that I spend some time working on is thinking about sort of the environmental variability. How can we get the ocean physics right, and get the ocean physics right in what context? So this is just a plot showing spatial and temporal heterogeneity of transport predictions using the global HICOM ocean model, releasing bunches of particles off the coast of Japan. You can track sort of the maximum eastward transport based on the start location. You can see there's a lot of heterogeneity in there, and spatially there's also quite a lot temporally. So this shows if you did the same set of simulations the day before, and you subtract out the maximum longitude obtained by particles released here from the ones the day before. So this is a change in longitude of, upwards of 30 degrees, plus or minus in a given direction, from a spot. For context, I will say that this is tracking particles over a 2-year period, 2.5-year period. So there's sort of a lot of room for dynamics and variability. But, this does, I think, nicely highlight that, that for thinking about organismal movements, sort of broad arrows in your textbook diagrams aren't always strictly going to be the most informative if we're talking about, being able to mechanistically predict those movement steps. That we're going to need relatively fine data on ocean current conditions if we're going to achieve that. And so thinking about, how do we do this, what's sort of the sensitivity around our ability to measure ocean currents, is something that is an important thing to do if we're going to get the biology right, if we're going to understand sort of the ecosystem level processes. So here's just an example of different ways that we can look at ocean current conditions. So these are showing sort of velocities of particles in HICOM and a drifter from the global drifter data set. And depending on how it is you're trying to characterize the ocean currents, so in this case we've looked at HICOM velocities. These are those purple lines coming off this sort of sequentially, these daily intervals across the trajectory of that drifter. And we can see that using that sort of method, you can follow the speeds reasonably well. There's a strong correlation between the HICOM predictions of velocity and the drifter's measurement of that velocity when you go about it that way. But simply releasing a cloud of particles at the same point and expecting them to characterize a individual's movement, in this case an entirely passive one, you see that it doesn't always do such a good job. So you need to, I'm not really telling this to you, more to me, I need to think a good bit about how it is that I'm trying to represent the ocean currents. Not all approaches are created equal, and even in sort of an optimized approach for trying to do it, taking great care in lining up ocean current measurements and the sort of the steps of our drifter organism, there's still plenty of room for improvement, and there's certainly plenty of room for assessing the sensitivity of our ocean current measurements. Now, I got hired on here, I think, to focus on a lot of the biology and making these links to ecosystems, how ocean physics can give us insight into ecosystem level processes. And so what I'll try to do is spend sort of the bulk of the talk then sort of giving a nod initially to getting your ocean physics right, but thinking about how we can apply those ocean physics models and tools to understand the ecology of marine organisms. And to acquaint you briefly with some of my ongoing work on sea turtles. Sea turtles are a neat study system for this, in part because you get to have this comparative analysis within the life cycle of a single organism, where you have so turtles lay eggs on sandy beaches, and then the little ones disperse out into the open ocean. They're not particularly great swimmers. Ocean currents are probably a dominant factor in their movements and distribution. As they age, they eventually come back into neritic waters, and that process may be a combination of their behavior and the sort of the gyre systems that are sort of characteristic of ocean basins. But then as they age, become older, they become quite skilled, it seems, at going to specific locations at specific times. And so with this single taxa, we have sort of at our disposal the breadth of the movement spectrum. That notwithstanding, they're also of some management concern as well, given that they're protected species and we have a lot of laws and regulations surrounding what can be done with these turtles, what can be done in areas where these turtles are, and so finding out about their movements and distributions is of concern from that regard as well. So so starting with thinking about hatchling dispersal, we certainly can make predictions about where they are going to go based on ocean circulation models. So these are some predictions based on global HICOM again over 3 different years, releasing from the coast of Brazil here, about 5 degrees south. And I didn't show you the interannual variability, but I'll tell you there is some. In some cases, you end up with turtles, or not necessarily turtles, excuse me, particles moving into the northern hemisphere, some moving into the southern hemisphere. And unlike, say, in many places, say like Florida, where the flow is going to be directional in a unimodal sort of sense, north and out, this seems to be a potentially nice scenario to look at this environmental variability in a very broad sense, in a very dramatic sense, in terms of the potential for turtles to end up in opposite hemispheres. So we might like to know, where is it that the turtles are actually going, and how might they get there? So this project is funded through NOAA. University of Central Florida kicked into sort of the brains behind the tagging operation was Kate Mansfield, and we had a nice collaboration with the Tamar group, who does turtle stuff down in Brazil. And what we ended up doing was the Tamar folks raised a bunch of turtles. Kate put some satellite transmitters on them using some hair extension glue and released them out there. They were nice enough to put some drifters out there at my request, so we could make some determinations sort of measuring both the turtles and the currents offshore of Brazil. And what this shows you is, here's a release of the turtle, release of the drifter. Note we have slightly different scales. The dark lines is the track of the turtle. The dark lines here are the tracks of the drifters. The light green is particles released from a HICOM model at that same time and location as the turtles. Again, to give us some context of what the currents were doing along the track. In addition to this bit of information from the drifter, the dots are plotted at 2 days apart. And I think that's going to be the same across the things I show you. And so here we have a turtle that is clearly going north. I think that's Barbados. Making their way into the northern hemisphere. The little drifter starts off going north, but it quickly runs ashore. Interestingly, I suppose, the turtle did not run ashore. Here's another snapshot of turtles released at a different time of the hatching season. In this case, the turtles went south, the drifters went south. We have a nice agreement there between HICOM and that drifter. We have certainly southward flows for that turtle as well, maybe a couple more gaps, potentially indicating some swimming behavior going on. Then this is sort of the third movement type that was sort of broadly characteristic of our turtles. Here, ocean currents, I think you can maybe see, well, certainly you can see we have sort of this circling motion. We have sort of an eddying area, not highly directional. And you can see that here in the differences of the directions of those HICOM trajectories. And in this case, the turtle track is not particularly well represented by the ocean currents, whereas the drifter is. What certainly seems to be the case is that the variation in ocean currents is an important aspect of where turtles go. So when ocean currents are generally going northwards, the turtles seem to go northwards. When the ocean currents are generally going southwards, the turtles seem to go southwards. And when the ocean currents are sort of eddying offshore, the turtles seem to stay in that area. And that seems what the currents are doing within the first, say, 10 days of release pretty much set that in terms of once the turtles, within that 10-day period, we don't ever have turtles that were going north suddenly going south, or vice versa, or turtles that had been hanging out in this area suddenly going somewhere else. But what is sort of interesting in this is that we see at these different releases that we did, which sort of coincide with early, mid, and late of the hatching season. What we see is that, one, there's significant variation in the ocean currents. There's also significant variation in the turtle's orientation behavior. So subtracting ocean velocity from the track velocity, the turtles seem to orient differently depending on the ocean state that they are in, which is somewhat surprising. It was surprising to me. My anticipation would have been that the turtles have a relatively sort of canalized set of behaviors. That's what they do. The ocean induces variability on that behavioral program, but the turtles themselves would always behave in approximately the same way, and it would be the ocean inducing variability rather than some sort of combination of the two. Within that, though, I will say that those calculations of swimming behavior are inferred from the HICOM model, and while we see that there's variation in orientation among those sort of three behavioral types, there's not a variation in swim speeds. So the turtles are swimming the same speed. The errors that we see comparing HICOM to drifters are the same order. And so I don't think that it's likely to be an artifact of the methodology, but rather something that's actually real. So how is it that you might be able to change your orientation? How might you set your orientation relative to changing ocean conditions? You're in the middle of a current, it's moving, you're in it. What's your frame of reference to know that you are doing something different or going somewhere else, that you're going north or you're going south or you're going east versus west? And in my earlier days of research, I was looking into how animals use the Earth's magnetic field to perceive both direction and spatial location. And so turtles are among the animals that are capable of deriving both compass information and what we call map information from the Earth's field. Compass is pretty straightforward, I think. As you can imagine how you could do that with a compass. You can feel the alignment of those field lines. That pulls, you have the receptor that can feel that, and you can say this way is north, and I'm going to set a heading X degrees off of north. Map information, so the Earth's field is a vector. There is spatial variation that is predictable. So for instance, the strength of the magnetic field is weakest here along sort of the magnetic equator, and as you go towards the poles it gets increasingly strong. So you could keep up with those sorts of things, and in principle you could know roughly where you were and which way you might want to go. And so it's way back when, in 2011, I showed that turtles were capable of extracting both latitudinal and longitudinal information from the magnetic field. It's a little hatchling from Florida. If you put them in a lab setting and expose them to magnetic fields characteristic of these two locations, they swim on average in opposite directions, showing that they can discriminate that and use that for orientation information. More recently, though, I've been collaborating with a fine young man at the University of Miami, not on turtles, but with zebrafish. So working with him and some colleagues at the University of Naples, we're sort of asking the question, does the magnetic field influence animal orientation relative to a current flow? That's sort of what we were interested in the case of that Brazil currents situation, where the behavior of the turtles seemed to vary in response to the currents. This was a very artificial sort of lab setting. We have a flow chamber here, flowing water through that tube, putting a handful of zebrafish in there, just sort of your lab rat of the fish world, and presenting them with a magnetic field through these coils here that was in different directions with respect to the flow of the water. And it's a simple experiment. It's something he did as part of his master's project. But the short of it is that this is the zebrafish orientation into the current. So the Rio Tactic Threshold. So the Rio Tactic Threshold being how robustly and at what point the fish will orient into the current, sort of beyond random, that you get all of your group of fish pointing into the current. And that is actually modulated by the magnetic field direction relative to the current. I won't go into too much of the specifics here, beyond that it is conceivable that the movement of water, or of an organism in water, relative to the magnetic field could provide that frame of reference that would be useful for understanding where you're going within that flowing environment. And this just shows we didn't get much information out of the animals in solitary conditions. We did when things were normal. They were with 5 or so of their friends, and then if you treat them with cobalt, that reversibly disrupts neuromasts along their lateral line, and it results in an opposite pattern of the magnetic field effect. In any case, we can certainly say that that I think gives some indication that organisms, again, are capable of responding to these changes in current, and it points to a potential mechanism to look into as far as how they might go about doing that. But that's all been, I guess, very heavy on the sort of the mechanisms of movement, that movement step process. We're also interested on the ecological side of things. So what are the implications of organismal movement mechanisms? And so this is a project that I worked on with a PhD student out at Deakin University and some folks within NOAA, Pacific Island Fisheries Science Center, to tag and track young-ish loggerhead turtles from this location. So this shows about 40 tracks of turtles and the date of duration. And so what we're interested in with this particular set of questions, so we're releasing all 40 at once. So instead of looking at sort of this temporal variability like in Brazil, we have a big cluster of turtles. What does a big cluster of turtles do released from a single point? And so with that, we developed sort of these movement transition matrices for turtles, for drifters using the global drifter array, and using HICOM. And what we could see from that is that the movements of the turtles, one, differ from the movements of drifters. So if you put a turtle in this turtle transition matrix here and run it for 100 days, they end up with a different distribution than if you did the same thing with the drifter matrix or the HICOM information. You can see, looking at different points along that, you see differences, looking at divergence rates between the turtles and each of these two other parameters. You can see in some locations turtles are very different than what ocean conditions are doing. Other places their movements are better predicted or better accounted for by the movement of the ocean currents. And in others, again, it seems to change somewhat. What we can see from this, so looking at, what does that mean for the turtles? So this shows the sea surface temperatures encountered, sort of the proportions, and the chlorophyll A, so sort of, again, a metric for the base of the marine food web. And what we see in particular is that the movements of the turtles result in some cases of them reaching areas that have higher chlorophyll concentrations, and that also in that same instance are somewhat cooler waters that might have some benefits in terms of their metabolism and growth. They're ectotherms. They need food in appropriate temperatures. In cooler waters, their metabolism will be lower. They won't burn through energy as much. They will, and if there's food around, that might be a decent place to be for a bit. So the short of that set of analyses was just to see, how it is that the turtles' movements are influencing sort of these processes that might be important for their ecology, namely sort of growth and survival. So on the same sets of questions, we can move a bit more applied. So we can think about what are the management implications for organismal movement mechanisms. This shows a plot of green turtle nesting beaches and foraging grounds. Nesting beaches are these white squares, sort of the number of nests are shown. Bigger squares have more turtles there. The green dots are known foraging sites of the turtles, and in what you might expect is that with that oceanic dispersive stage that we have in these turtles that might last a couple of years, there's the potential for turtles from this location to end up very far from its home. There is also the potential for, as it's going out, to run into maybe some trouble along the way, run into some fishing gears. And if you are wanting to manage this whole population, then what you would need to do is know where the turtles are coming from that are, say, if you have a fishery here, what proportion of the turtles that are in this area are from each of these different locations. What your management recommendations might be could differ. If you are consistently getting turtles from a robust and healthy population, maybe you're not so worried about a turtle snagged on a line or two. If you are mostly getting turtles from a population that is on a downward trajectory, then maybe you should have a different set of management conditions. So here's a recent example working with some folks who were looking at lots of green turtles coming into Texas waters. This is a these are the numbers of turtles from strandings along Texas. This is their size. This 40 centimeter cutoff, or not necessarily cutoff, but this 40 centimeters where the bulk of the turtles are, shows you that these are primarily the younger stages, that dispersive life stage that they're finding here in Texas. And well, in Texas there had been a lot of green turtles, like 1800s, early 1900s, lots of green turtles. Then not so many green turtles. They all got put in cans. And in the late 90s, early 2000s, they started showing up again in pretty decent numbers. The sort of a question was, where are they all coming from? Whose turtles are they? They're not Texas's turtles, for sure. And so what people did was use some genetics techniques. So you have these disparate nesting sites, and because turtles tend to go back to their same nesting sites, you have populations tend to become genetically distinct. And so you can use genetic markers to infer origins. If you have some turtles out in the ocean, you can say what's their likelihood of coming from a particular area. Unfortunately, from this 2013 paper, the relatively poor resolution of the genetic markers resulted in a bit of uncertainty. There's our bit of uncertainty. Those are some awesome confidence intervals, aren't they? So that said, I mean, they clearly showed that there's sort of, they call it Gulf here. They mean these three nesting sites. So they're probably from, the Western Atlantic, probably Northwest Atlantic. But whether they're from Florida or Mexico is sort of a crapshoot, pretty much up in the air. Mitogenomics and movement models to the rescue. If we look at the ocean current conditions here, so in this case, I released a bunch of particles in global HICOM and backtracked them to see where they would come from to reach that final location there. For the years, this is just showing 2000, but for the years where those samples were obtained, 99 through, I think, 2002, we saw a lot of potential for Mexico and in the Caribbean as far as sources. We did not see any potential for Florida, for the Florida nesting aggregation. And so then this shows just from those models the predicted contributions of green turtles to Texas during that time period, and nothing from Southeast Florida, which was the primary nesting site. A lot from Mexico, a little less, some from Costa Rica. Venezuela is sort of an interesting one to think about, in that there aren't genetic markers for the Venezuelan population. They haven't been sampled for it. So whether that is in agreement or not remains to be seen. But with the new mitogenomic analyses, so mitogenomics is looking at the entirety of the mitochondrial genome rather than, say, just a small 384 base pair kind of thing. So with the new mitogenomic analyses, you can see that the error bars have come down a little bit. So this one's Costa Rica. This one is another, these are the Mexican nesting areas. And you can look at the relative rookery contributions based on just the genetic sequences themselves, when you can also weight those by the population sizes of the nesting sites, and you can weight them by the currents and the population sizes. And you can see our error bars get smaller with taking into account currents and population size. And where's our Central Florida, South Florida, those error bars are sort of that appropriately small stage given what we know from the currents. And then here's just a pie graph showing the proportion of haplotypes, that those sort of different genetic markers, with purple primarily in Texas and purple entirely measured in this Mexican rookery here, whereas there is no purple on the east side of Florida, so we can sort of rule that out. So then, the turtles in Texas are almost certainly of Mexican origin, and therefore the problems that they face, as we're stranded turtles, of course, will be of concern for these. Somebody else's problem, right? No, but they aren't affecting the East Coast turtles, which are on a upward and happy trajectory. So we've hit a bit on mechanisms of movement. We've hit a bit on the sort of ecological implications and management implications. So now I'd like to talk to you a little bit about where I see us going with this, so that the emerging research directions. And what we're aiming for, I guess we'll start at the top and work our way down. So this ecosystem-based management of spatially and temporally variable resources. It seems like a good goal to be able to take that into account and manage that. And here's my view on how that could be done. So taking sort of the movement ecology information, so that mechanisms of movement steps, which feeds in to give you information on not just information, but the mechanisms that drive distribution, that shape the biogeographic patterns, the population connectivity, the phylogenies of metapopulation dynamics and things like that. And those feed in to give you some information on sort of a dynamic biogeography that's not static, that's mechanistically linked to behavior, to the underlying environment, so that it's changing over time and you're able to take into account that possibility. And so one of the, there are sort of two main areas that I'm working on this. One is in coral, which don't have very much movement, at least not for very long. So they have a sort of a short larval dispersal stage, and then they are hard and stony and sit at the bottom of the ocean. So one end of the spectrum. The other side we're looking at is pelagic sargassum, which is in constant motion. It's always going somewhere and until it beaches. So for the coral side of the work, this is being done as part of NIMP's Protected Resources Quantitative Toolbox project, and it's a joint effort to have these three aspects. Predictive species distribution map tool, so thinking about the substrates, predicting where those coral will be, what's their habitat. That's Shay Veeman's work. This population assessment and spatial demographics is being done by Dione. And then I'm looking at this connectivity and recruitment model question, so the movement sides of things. And so, as I suppose I briefly described the life cycle of the coral, you have these coral, they spawn, their larvae go up into the water column, they drift for a bit, they find a place to settle. And they're important. Lots of species use coral reefs. There's concern about the implications of global change and land use causing problems for corals. And one of the things that we need to think about in terms of sustaining corals is their recruitment. Will they be able to replace themselves as they have for millions of years? And so what I'm trying to do with this is to, within the Florida Keys, identify regions that allow for the recruitment of larvae under sort of recent and hopefully future conditions, and then make predictions as to how distribution and abundance might change in response to the changing climate, in response to changing recruitment dynamics. So asking questions like, to what extent does recruitment in the Florida Keys depend on external sources? There's just an example of backtracking to see the contributions from Mexican northern Caribbean coral sites. And this also involves questions about which locations are most likely to receive coral larvae as inputs. This shows, this is log scaled and assuming the coral larvae are at 0 meters depth. Anyway, this shows you estimates of where coral larvae should be coming in, where you, at a regional scale, where you would expect them to be most likely to recruit. These might be most likely then to be replenished down here, whereas these might not. We can also ask about which locations promote intra-regional connectivity. So this shows if you release a virtual particle here, and what is the distance it will travel before recruiting, given some assumptions about its position in the water column, its period to competency to settle, and those sorts of things. We can also ask questions about which locations in the Florida Keys are responsible for producing the new recruits. So this is the probability of this spot producing a larvae that will find its home somewhere along this track. And we can see that there is certainly some variability in those predictions depending on where the larvae are predicted to be in the water column. So at 0 meters depth, their recruits are less likely to originate from these locations than if they're at 2 meters depth. But we can also see even sort of greater sensitivity to some assumptions about the biology. So a 3-day competency period versus a 9-day competency period. This means they can't settle until they're 9 days old. This means they can't settle until they're 3 days. You can see quite a different set of scenarios. So again, the biology and the ocean physics sort of need to go hand in hand in terms of making use of these this framework for studying it. Certainly requires some, can do a lot of sensitivity analyses, but it'd be nice to narrow down those confidence intervals with some in-water data, in-lab data. On the sargassum side of things, this is a project that will be involving a host of folks, a number of you in the room. Thanks for helping out. And we'll see who wants to pay for it, but I think some elements of it will be able to continue. So we have this interesting situation. We have pelagic sargassum. It's this Lagrangian ecosystem. It is a species. It is, it's an organism, but it's an ecosystem as well. And it's important for fish. It's important for turtles. And so understanding the movement dynamics of it in the context of the organisms using it, the animals using it, so for instance for fisheries, for protected species, is of some value. So, pelagic sargassum is not just an ecosystem, it's a bit of a problem. So I think this is Sierra Leone. It's just a huge pile of sargassum. In recent years, there's been a number of beaching events in the Caribbean and in Africa, among other places, where this sargassum has piled up and has caused problems for sort of coastal management, for tourism and things like that. And so it'd be nice to know when that's going to happen, how that can be potentially mitigated, give people a heads up on those sorts of things. So studying the movements of it and the mechanisms that drive its movements would be of some value, both on the sort of understanding ecology as well as some of these sort of very tangible human dimensions. So what we're working on is pairing satellite data. This is taken from Chuan Min Hu, his work, using satellites to observe ocean color, make inferences about where sargassum is, comparing that with hopefully some comparative genomics data, where we'll have not too dissimilar from those turtle studies I was telling you about. We'll have, we can collect data from sargassum in different locations and get in build some inferences about genetic connectivity. And then also doing things on the more mechanisms, sort of those movement steps, by pairing some drifter and telemetry experiments with ocean circulation modeling techniques to show where to adequately describe the movement of sargassum sufficiently that it can then be modeled using products like I've been talking about all day long, so backtracking, global HICOM, those sorts of things. I think this is a valuable framework. Movement ecology certainly, I think, is it's on this side. I think it's foundational to getting to this red part. As you can see, everything here is sort of building on this this cornerstone of organismal movements. And with that, I think it's worth thinking about on the movement ecology. So here's our movement ecology box. Here's what's inside of that movement ecology box. So this is back in 2008. This is just sort of looking at what it is, what are the proportions of research studies, the projects that are going on in terms of studying movement ecology. And what you can see is that certainly a lot of focus is on how external factors and motion capacity feed into movement. What's lacking a good bit, those lower numbers, are these questions in terms of the organism's behavior, the organism's sort of physiology. And so what I would say is that probably to make progress in this area, what needs the most progress within the movement ecology part is actually going to be understanding how organisms are making their movement decisions, what motivates them, you might say, to behave and move in the ways that they do. So with that, I will conclude with telling you that I do think the movement ecology framework as part of that larger whole is well suited to address questions about how environmental conditions influence biological, ecological processes, and then also that by using that framework, we can better understand the interactions between biology and the environment and make better informed management decisions related to marine species and ecosystems. And that sort of more broadly, on the how we go about doing this, I think multidisciplinary studies combine multiple approaches of observation, experiment, modeling, ocean physics, genomics, behavior, a wide gamut of fields are going to become increasingly necessary to be able to address the most relevant management questions that I think NOAA and sort of science in general is being tasked with. And so with that, I would be happy to take any questions. Thanks. Yes, Paul.
On the Brazilian example,
Yes.
the turtles, did you have genetics? Any genetic information with that?
Oh.
Corroborate that at all?
So on the tracking the little turtles,
Yeah.
so far there has been, so from 1998 there was an errant Brazilian haplotype that was in the Azores, and I think people said maybe it came from Brazil, but maybe it's just a rare haplotype in the North Atlantic that hasn't been observed yet. So I think that it's scant, but yes, I suppose there is some genetic evidence that would be consistent with northward movement of those turtles.
And presumably those ones that swim north, there's some genetic component to that behavior, and presumably you collected samples.
So those, So I think what you're thinking about there is sort of a functional genetics question, which is a good question, but I don't know if we're where we need to be on sea turtle genetics to address that. So what folks have mostly looked at are these, like as neutral markers that tell you maybe where it came from, but it doesn't. They're not looking at things that code for orientation behavior or that kind of thing, unlike what's been done in say, migrating birds or butterflies or other species. But yeah, I mean, it would certainly be interesting to see. So on the genetics thing, though, I don't think that it is just that there's a north-going subset of the Brazilian population, because some turtles that were hatched in the same, or they're from the same clutch, but were put out at different seasons. So they were put in the ocean, and it has to do with at what size they grew to when they went out, I believe. But the short of it is, you had turtles from the same mother that were put into the ocean in November or May or maybe October, sort of that, and they didn't do the same thing. They did what the ocean currents dictated.
Do you have enough like number of turtles that are found somewhere to have a link between changes in ocean currents in location or intensity and the assessments of the number of turtles that you find somewhere? Is there enough observations of turtles?
There should be. There should be. It has not been done. That is something that Paul, who just asked the first question, and I have been kicking around about, like with the strandings, for instance. There's this huge stranding database that goes back from, when is it, late 1980s or something like that, to present, that just people who find turtles washed up tell you about them. And so you might have regions that, by month, you have X number of turtles over that time period. And so yes, it seems like you could correlate changing ocean conditions with variability in turtle inputs into that system. It's definitely on our radar and sort of being done in my free time. But yeah, it's a really good idea. There are problems in accessing those data about the turtles. The ocean currents are pretty easy to get.
That's it.
A combination of the states state governments and the National Marine Fisheries Service. And yeah, certainly volunteers primarily are the ones or the public who report them, who houses the data are states and NIMS. Yes, Lou.
Hi, thanks for a great talk.
Thanks.
I have two questions. One, what is the Crucible Cymru, and why is Wales funding your research? And
that's a good question.
two, this Rio Tactic or Rio Taxi is really interesting. Has there been much research done on survivorship advantage or ultimate evolutionary advantage to having that capability for turtles or other organisms?
So the first thing, I pulled this slide from another talk that had some about eels on it, and the Welsh are interested in eels. Like this guy here, Tom Quinn, he didn't do anything. He's a great guy. He's at the University of Washington. He contributed some on my salmon work. So, yeah. But to your other question, the adaptive benefits of being able to do that. So certainly on the Rio Tactic question, being able to orient with respect to a current is, in theory, very valuable, right? Because then you can maybe minimize swimming costs. You can set heading to transit appropriately and quickly, or slow down your movement out of an area if you want to stay there. So there's been a lot of theory about how animals should be moving in fluids to optimize whatever, essentially retention or dispersal. But there has not really been a mechanism for how you manage that in the open ocean. Rheotaxis in a river is pretty straightforward. If you have a bottom, if you have visual contact with things that are stationary, that's a reasonable thing. But sort of the idea in the open sea is there is no frame of reference to tell that you're moving with the current. Has sort of been the primary vantage point for decades. That's not to say people haven't floated, sort of weird ideas about detecting Coriolis force or looking at the North Star, various things that might, again, floating them as ideas to look into. But as far as the, having the magnetic field as a static reference point could be potentially valuable as a way to do that. So if the field is stable on the scales that we're talking about, and so if you can detect the field and you feel and then you can feel the field changing as the ocean moves you, even if you're not feeling the ocean itself. That little laboratory experiment is suggestive of a potential maybe possibility. I think it's a ways away from showing that with any sort of definitive. I mean, it's in zebrafish. It's in a lab. Blah 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 blah. But I do think it is sort of I don't know. Is an interesting enough possibility to me. I thought it warranted inclusion. So the adaptive benefit would be having a stable frame of reference to gauge drift by and modify your headings accordingly.
It suggests to a simple physicist that these turtles have some sort of learned knowledge of the structure, the geographic structure of currents. But that sounds far-fetched.
So certainly with the work, the orientation work that I've done with turtles, with salmon, and with eels, the orientation responses all seem to be adaptively tuned to consistent ocean circulation features. So like gyre systems. And they seem to reduce variability. So if you simulate those behaviors in an ocean circulation model, what invariably happens is the relative to, say, passive drift or random movements or those sorts of things, the variability in the migratory route is reduced. So it seems to make their world more predictable and it does seem to be tuned to sort of average ocean conditions. To what extent that's learned versus, I think we will probably get into an issue of semantics and what a physicist means by learned versus a psychologist or a biologist, right? And so are we talking about genetic? Yeah. But yeah. Yes, ma'am.
So there's a few, I work at the National Fisheries Service, and there's a few things that struck me during your talk that are kind of obvious collaborations that could exist. And one is that the International Commission for the Conservation of Land Tunas is very interested in establishing kind of the susceptibility of turtles to fishing gears. And so we have a lot of information about nesting sites and things like that. And we also have a long line simulator that kind of simulates the gear, the vertical distribution and spatial distribution of gear. But there's been little work to try to establish where you might put closures, for example, to protect certain life, stages of turtles. At least not for ICAP. And the other is in the Gulf Council is very interested in establishing the role of sargassum for some of their managed species. And one in particular is gray snapper, gray triggerfish.
Okay, yeah.
They would like to know whether the availability of sargassum actually influences recruitment of that stock. And so just a couple things that you mentioned. I can send.
How much information is available on gray triggerfish catch?
We have a whole assessment, so there's a lot of information available. What we don't really have right now is a time series of sargassum habitat in the Gulf. And that may exist, but we don't currently possess it. So I can just drop you a note with a couple of people who might be interested in collaborating with you.
Okay. I talked to Steve Turner a little bit about some things in the Caribbean, but not about gray triggerfish or Gulf of Mexico stuff. That'd be, please do. Other questions?